All right. So, unfortunately, today, and this may take up some time to go over this, I have some really bad news to share with all of you. So, uh, essentially, uh, I'm a legal alien, which is a real funny phrase, to be honest, to me. But anyway, I'm here on a H-1B visa, and that visa expires midnight tonight. And since I started working, or since I joined Texas A&M San Antonio, um, I've been pushing for them to sponsor me for a green card, which is some of you probably know what that is. But essentially, it's a change of status to permanent residency, which means as long as I live here for, or me and my family, to be completely accurate, as long as I live here for, I don't know, 9 months out of the year, we get to stay as long as we want. Still can't vote, but that's a different issue. Anyway, so essentially, the university, for whatever reason, which I still don't really know, screwed up the first attempt to do that, and then dragged their heels on the second attempt. And basically, what it means is that I do not have a work permit yet. That was actually what I went for my biometric stuff with my wife last week. So what that means is that I can't do any work, right, as of midnight tonight. And in terms of work, that means anything that can be construed as work. So I cannot teach, I can't do research, can't do any service work, I can't answer emails, I can't answer my phone if that is to do with anything related to work. Because if I do, then I jeopardize my green card application if it's found out, obviously. So what that means for all of you is that on, well, I've got to tell this to my other section, genetic section as well, which is going to be even harder because I'm not going to be able to speak to them. But you will have a replacement teacher for your next class on Wednesday. That's going to be Maurice Penny. Don't know if he's doctor or not. Um, and he will be essentially doing what I'm doing now. I'm going to be meeting with him. I found out that he was going to be a replacement on Friday, which is a little late. And he's also taken over for the labs as well. So some of you already have Mr. or Dr. Penny. I actually have to find out which, given his correct title. For lab, that will continue. If you have me for lab, then you'll also have Mr. Penny as your lab teacher as well. For Wednesday lab participants, I still need to sort out the student pairings. That's on my list after today. I had other stuff that I had to do this weekend, unfortunately, which is ironically tied to me keeping my job, which seems like a funny thing to think about right now when I can't do my job. Anyway, he will be sending out, so I'm going to be meeting with him this afternoon by Zoom. He's had the weekend to go over all of the course materials. So all of the material that I've posted for you, essentially, PowerPoint, schedules, audio recordings, things like that. I'll also be providing him with sample exam questions for the different sections that we're going to be doing, which he can choose from and then add his own to, depending on what subjects he covers and how. The aim will be that he will essentially continue what I'm doing. Given these synchronous Zoom sessions, he will send out a link to a new Zoom session. Yes, Lauren, that's a good question about labs. The kind of the pattern that's already laid out in terms of partners and who will be attending when will continue. The only thing that I need to do for the Wednesday section of the lab is get that document out to the students so that they have a record of that for other reasons, which I don't need to go into here. So they're a little bit behind the curve compared with the Thursday lab as to who's doing what and when. But yeah, the exact same format will continue. Half the students attending one week, half the next week, and sharing data and information between them. So he'll obviously also need to send out links for the Zoom links for the labs. I'll try and convince him to do the same thing that I was intending to do. The schedules stay the same, so everything that's laid out in the syllabus and the schedule will stay the same. So really, the hope is that nothing will change other than the person that will be teaching you. And ideally, the quality of the teaching will also stay the same. I don't know Mr. Penny well enough to give you any kind of feedback about that, but you know, I mean, it is what it is. We can't really make any changes anyway. Our choices were limited as to who could teach this. So if there are any questions, I'm probably going to be inviting a deluge of emails, but if you have any questions about this, obviously now's a good time to ask them. If not, then send me emails, but I can only respond to emails today. From tomorrow morning, you know, essentially midnight tonight, I cannot respond to any of your emails. Any questions have to be directed to Mr. Penny for him to answer. I can't even converse with Mr. Penny about any questions that you may have that he can't answer, unfortunately. So hopefully he'll be able to lean on Dr. Page, who is the genetics professor before me, for any of those kind of trickier questions. The big downside is I do not know when I will get my work permit. So it is quite possible, hopefully not, because we'll be in pretty dire straits if it were. It's quite possible that I won't be teaching you again unless you take this class next semester. So I don't know, because I've never done this. I've never applied for a green card before, and I don't know how long the process takes for getting a work permit. I expect to have a green card by this time next year. Hey, that'd be great. Won't have to worry about getting kicked out of the country anymore. But I don't know how long the work permit takes to arrive. So it could be next week, in which case you'll just miss my lovely beard for only a few days. It could be by Christmas. I don't know. There is no knowing how long this takes. My expectations, because I've kind of run out of hope, to be honest. This has been going on for 3 years now, right? I joined Texas A&M in August 2017, and this has been kind of a pretty heavy burden to live under for the last year and a half, really. The application for like the start of the application was last May, so May 2019. So pretty much since then, we've been running against the clock trying to get this done in time, and it hasn't. So I don't really have an awful lot of hope that it'll happen anytime soon. I'm kind of out of hope, to be honest. And my expectation is going to be between 1 and 3 months before I get the work permit. And yeah, thank you for your well wishes. It's much appreciated. We're now going to be on ramen and water for sustenance. I don't know. Actually, I'm going to start trading car mechanic works for food. That's my plan. So yeah, I'm really sorry. I am abjectly sorry, to be honest, about this situation, because it's not just about me, but it's also about all of you. I'm very sincerely hope that this does not affect your ability to learn this subject and succeed in it. Unfortunately, there's nothing I can do to help you other than what I've already done. But I will be, well, I mean, I do need to apologize, Erica. This is a monumental screw up, and I doubt that the university will be apologizing to you, so it's my job to do so. Yeah, so I'll do my very best to make sure that Mr. Penny, or Dr. Penny, is, oh yeah, I'm sorry, Lee. It's not a nice, it's not a pleasant situation to go through. My hope is that he'll have everything that he needs to do the very best job that he can. And he seems like a genuinely decent guy, from the little that I've interacted with him. Pardon me. That he'll be able to do a good job. And we'll just see how it goes. Fingers crossed it's not going to be too long. But we'll just see. I'm going to be sitting here busy doing other things, which are obviously not work related. Ha, never. Can't do that because that's not legal. But I'll be keeping busy doing other things until I get to come back. And as soon as I get that piece of paper in the post, whenever that might be, I will be picking up the reins again the following day. So there's no delay in terms of when I resume my work. It's literally the day after I get that receipt notice from the USCIS in the post. Does anybody have any questions about the looming situation? All right, well, if you do, again, get those to me today. Mackenzie, lab will be in person for those who are coming this week. So for half of you, yes, lab will be in person this week. This week is corn genetics and chi-square, which also more Dr. Penny, Mr. Penny, I really need to find out which, will be teaching you in lecture as well. So that's the whole probability, statistics and probability in genetics topic. But yeah, so for half of you, you will be going into lab this week. To come in person, essentially, you in Wednesday's lab, Oliver Tooby? I can't remember which. Okay, yeah, so ideally before lunch today, I've got basically my brain just dribbled down my ears yesterday when I was trying to get this together. But after class today, I will put together that partner list so you know who will be coming in on Wednesday and who will be coming in next Wednesday. Yeah, everything stays the same, Delani, if I got that pronounced right. The room for the lab stays the same. What you'll be doing in the lab is going to be exactly the same. Mr. Penny and I have already been organizing, so he knows what he's going to be doing every week. Pardon me. So everything stays the same. For Christina, the Thursday lab, the list will stay the same. I think, so the reason why the Wednesday lab list is still up in the air is because I had some issues with announcements on Blackboard. That when I actually asked for lab partners, that announcement didn't go out. So I gave them a little bit of extra time to sort that out. But the Thursday lab, the partner list stays the same. In terms of who goes in on Thursday, you can organize that with your partner. So whoever you've been partnered up with, I've given you, essentially, this half goes this Thursday, this half goes next Thursday, and they alternate. But if for whatever reason between you and your partner you wish to flip that round, I don't mind, or well, Mr. Penny won't mind, and I'll talk to him about that. As long as you maintain that order for the rest of the semester. So I've just arbitrarily put you into this Thursday and next Thursday. Actually, I think it's mostly alphabetical because I was like, this half of the class roster goes in this bit, and this half goes in this bit, because that was easy. So if for whatever reason, scheduling or daycare or jobs, whatever, you want to flip it around, that's totally fine. Doesn't really matter. Labs are on Wednesdays and Thursdays for me because I have 2 sections of labs. So if you're in the Wednesday lab section, you're still in the Wednesday lab section. I'm just fielding questions from students from both sections, which is why it might seem a little confusing, Raquel. And obviously I don't get to talk about this stuff with you in lab because labs are on Wednesdays and Thursdays, and I'm off, at least for this week for sure. Yeah, so the hope, anything that hasn't been fixed yet, and that the only thing I'm aware of that falls under that category is the partner list for Wednesday's lab. Everything else stays exactly the same. So you don't have to worry about a new syllabus or a new schedule or new materials or anything like that, right? So any work that you have done already will count. All the work that you are to do will remain the same as if I were teaching it. The only thing that's going to change is that there'll be a different face on the other end of the camera, or a different person in lab teaching you. So hopefully what you'll get out of it in terms of knowledge, skills, understanding, and the like will stay the same. Obviously, I can't guarantee that it's going to be exactly the same as if you had me. Maybe that's a benefit. I don't know. Hopefully not. But the hope is that we'll continue exactly the same as if we were with me teaching. Just I won't be teaching you, at least until that piece of paper comes through the post, whenever that might be. And as I said, I'm not hopeful because that's all gone out the window. But one would wish that that was going to be within a few weeks. But I have no expectations that is going to be the case. It may be, it may not even be until next spring. I don't know. Hopefully not, because that would not be good on a lot of different levels. But I don't know. So my best hope is that I have done enough to get all of the material that is necessary for you to get the education you need in this topic is already available and provided, and it won't be dependent on who you get to replace me. That's the hope. And my hope is that he does not only meet that minimum, but also is able to, exceed it and give you what you need. But either way, hopefully it's not going to mess you up too much. That's the hope. All right. Any questions? Hey, sorry, got to remind my wife something. Coffee, please, and milk for Sophie. Now, got to make sure I got my coffee supply stable. That would be a tragedy. I'm already thinking about my coffee. It's not even coffee time yet. All right. Any other questions? Awesome sauce. Does anybody have any questions from Friday's lecture? Basic monohybrid cross, language, definitions, things like that. And I'll just get that up already. Where's that gone? Doop. Here we go. Let's see if I can. I'm sure you'll miss my wonderful British accent. I haven't actually given you much of a British accent. I keep using American words for stuff and everything. How is that part of your cultural aggrandizement, if that's the correct word? Come on, get out of this. There you go. No more how's your father. All right, so yeah, that's kind of weird order. Anyway, so essentially what we have left from Mendelian genetics is the dihybrid cross, right? So although I haven't laid these out formally, they're worth repeating again. So again, remember that we're making a bunch of assumptions. Whenever we do a monohybrid or dihybrid cross, we're making a bunch of assumptions. They're kind of baked in to these crosses. Some of them are fairly straightforward, like there is no selection acting on any of these alleles, right? So one allele is not favored over another, because you can imagine how that will affect the ratios coming out, right? If you know, homozygous recessive is lethal, for example, that's going to completely screw up your cross, and you're not going to get the Mendelian ratio out the other end. There's no new alleles popping up, because that would confuse things crazily. And sexual reproduction is both occurring and it's random. Also, generations don't overlap, they're discrete. P generation, F1s, F2s, right? So those are some of the kind of generic assumptions baked into these Mendelian crosses. And I can't stress heavily enough that they are always the same, right? That's their strength, right? They're essentially somewhat artificial constructs, right? Because very little genetics actually occurs in these kind of cookie cutter kind of way that Mendelian genetics is laid out. But that's their actual strength, because then you look at what comes out and see how that differs from what one would expect if these genes and alleles and so on were behaving in a Mendelian fashion, right? So it's really kind of like the base assumption, right? You know, we're assuming that all this stuff is going on, but in real life, a lot of it isn't. And the other two assumptions, right? So what do we have? Non-overlapping generations. We have sex is random, or reproduction is random. I mean, sex is sometimes random too. We have no mutations, no selection, right? So those are kind of the generic ones. We also have the law of segregation, which is, as you should already know, that which allele you get out of two is random, right? So those alleles segregate randomly into the gametes. And that's tied into meiosis, where you go from a diploid to a haploid cell during the process of meiosis, right? So there can only be one, ha ha ha. That's like my Guardian reference. Have anybody, the Highlander, anybody seen that? Hopefully someone has. Not that old. Anyway, watch the Highlander. It's you'll get the reference. Yeah, there you go. Someone. There's always a reassuring. It's like, oh, phew. Some people have watched the same films that I have. Anyway, so because of the haploid nature of gametes, then there can only be one allele, and so which one you get is random. That's the law of segregation. That applies to both monohybrid and dihybrid crosses. Right? We don't talk about it in dihybrid very much, but it's there, right? It acts the same, or it's assumed to be acting the same, right? It's probably the best way of putting it. The extra one for the law of independent assortment, I've already told you, it's up on the screen. For dihybrid crosses is the law of independent assortment, right? And so essentially, this is about more than one gene, right? And obviously the dihybrid cross is about two genes. And essentially what we're saying is that The law of segregation occurs at one locus, one gene, and it occurs at another locus, another gene, and those two don't affect each other, right? So the randomness of which allele goes into which gamete, right, is, you know, busy being random on its own over here. Pardon me. Just the same as the other loci is equally being random as to which allele goes into which gamete. Right? The two loci do not interact, right? They do not affect each other, right? And so, oh, that's a beautiful kitten. Wow, that would be intensely distracting. Is he trying to eat your finger? That's super cute. Oh, nothing like the sight of a kitten to cheer one up in the morning. Love, I've got one of my students showing me her kitten. Little black and white kitten. That's very cute. Thank you, Jane. All right, I'm going to just mask you with my, yeah, because I'd just be staring at the kitten the whole time and like, cat genetics. Anyway, gosh, shake images of kitten out of my head. And so this is a fairly big assumption, right? And so you'll see when we get to non-Mendelian genetics, and particularly when we go into linkage, that this really does not always hold true, right? And so this is probably the best example of that's cute. The best example of how Mendelian genetics is the framework, right? This is the basic structure which we then work off of, right? It is not, Mendelian genetics is not all inheritance genetics or transmission genetics by any stretch. But it's, you know, like, I don't know, like any scaffolding enterprise, right? You need to work off of something to get to those more complex situations, and Mendelian genetics really fulfills that need, right? So essentially, what we're saying is that we have two genes, and those two genes are far enough apart that they don't interact, right? And the easiest way about thinking of that is that they are on different chromosomes, right? And so if you have a gene on one chromosome, whichever allele you get for that gene in the gamete has nothing to do with genes on any other chromosomes, right? And so if we go back and look up here, if I can, here we go. All of these genes, all of the genes that control these traits that Mendel looked at, they are on different chromosomes. So the law of independent assortment really came about because either of accident or self-selection or whatever, but he was essentially working with every single trait being on a different chromosome, right? And so these traits, what allele you got for pod color, let's say, had absolutely nothing to do with what allele you got for seed coat color, right? Or seed shape or endostem color or whatever, right? So you could potentially have green peas in a yellow pod and vice versa, right? And these, if you had a dihybrid cross with these, you would get your standard Mendelian 9:3 to 3:1 ratio out of the F2s, right? These would not affect each other, right? And so that's a really important point to get across because actually, here's a question. How many chromosomes do we have? How many unique chromosomes, haploid chromosomes, let's say, do we have? Does anybody know? 23, right. 46 in total because we're a diploid organism. But in our gametes, we have 23 chromosomes. Does anybody know how many genes we have, roughly? I don't actually know exactly. I'm not sure anybody does. Does anybody roughly know how many genes we have as an organism? Plus or minus 1,000? No? Okay. we've got about 20,000 genes, give or take, right? And that kind of depends on how you define genes, and it depends on us knowing exactly what genes look like in order to be able to find them. But we've got about 20,000 genes, give or take, roughly speaking, right? So we have 20,000 genes spread across 23 chromosomes, right? That doesn't look like what Mendel worked with, does it? Right? So you are almost guaranteed to have genes or loci interacting, right, in ways which break the law of independent assortment, right? So reality, really whatever organism you study this applies to, maybe not bacteria, I don't really know much about those, but certainly any kind of eukaryotic organism that has chromosomes, you will have situations where the law of independent assortment does not apply, right? So it's a very useful tool, essentially, to see whether or not that is the case. Are your genes far enough that they act independently of one another, the genes that you're looking at? Okay, so if you want to ask that question, and it is a valid scientific question to ask, right? Don't do it much anymore because whole genome sequencing is so cheap, relatively speaking, that nowadays if you get a mutant, it's actually a lot easier and cheaper just to sequence the whole genome of the mutant and find out where that mutation is. But back in the day, when we were still using like, I don't know, chisels and stones to write out our theses, you can actually get, I don't know about other organisms, but I know for worms, C. elegans, you can get extraordinarily, hello, Luna. Luna the dog has come to say hello. Say hello, Luna. No, not interested. You can actually get strains which have like a fairly obvious nasty mutation on each chromosome, right? And so if you want to figure out which chromosome your mutation is on, you simply do a classic Mendelian cross and see which your mutation is interacting with which mutant chromosome, right? To give you a non-Mendelian inheritance, essentially, for that chromosome, right? So it does have practical applications, right? If you're trying to figure out whether or not two genes are interacting, this is the way you do so. So as with a monohybrid cross, and actually, you could do a tetrahybrid, you could do a heptahybrid. That would take a really long time. I'd have to like write that down on the floor or something. But whatever you do, you always have exactly the same generations. This is the P generation. I'm pointing at my screen, not you, obviously, pointing at my screen, see what I can see. You can do the same if you want. P generation is always true breeding, right? Purebred, whatever you want to call it. The parents are always homozygous. Not necessarily double homozygous dominant, double homozygous recessive. It doesn't actually matter. You can have them be homozygous dominant for one gene and homozygous recessive for another. The outcome is the same, it's only the phenotypes that will differ. And again, you can figure that out without knowing what the genotypes are simply by looking at the phenotypes of the F1s, right? Because if you have these individuals being crossed with each other, what is the genotype or genotypes of the F1 always going to be? That's a question. You get the eyebrows. That also means I'm asking you a question. Thank you, Mackenzie. Yeah, heterozygous, right? And which phenotype, dominant or recessive, does the heterozygous always display? Dominant, right? It's due to the law of dominance. So if we've been given the genotypes here, right? We've been given the fact that round yellow seeds are homozygous dominant for both. Wrinkled green seeds. That's kind of weird. I always thought green was like normal for peas. But anyway, wrinkled green peas are homozygous recessive for both. There. There's lots of sibilant sounds in science. Lots of alliteration as well. So in that case, what seed color and shape will the F1s have? These are the phenotypes. Yeah, exactly. It's going to be yellow and round. Don't forget, there's going to be two phenotypes because this is a dihybrid cross. We're dealing with two traits, two genes, and two phenotypes, right? So they will obviously be round, but they'll also be yellow, sorry, but they'll also be round as well. You have to remember that. Okay, so it's essentially what we got here. There you go. That's exactly what they'll look like. Now we have, so this is where things get a little complicated, and it's actually a little tricky for me to do kind of virtually, because this is where I need a big whiteboard to write everything out and draw lots of cats on. But essentially what you have to do now, and this is what you have to do for really any cross, is you need to figure out what are the gametes going to be. So you know that each gamete can only have one allele for each gene, right? So you're going to have two alleles in each gamete. You're going to have one for seed roundness and one for seed color, right? You're not going to have two for each of those because gametes are haploid, right? So what gametes can you get for individuals from the F1 generation? Talking about letters, right? We have heterozygous parents. Those are going to be the F1s. So they have a dominant and recessive allele for each gene, each of those two genes. So what possible gametes can you get from that individual? Oh, dang, Mackenzie, you're fast. Yes, exactly. So because you have two different genes and you have two alleles for different alleles for each of those genes, it's basically 2 by 2. So you get 4 different gametes, right? And so this is where we go back to the law of segregation, way back up in the PowerPoint. So because which allele you get for which gene in each gamete is going to be random, right? So essentially, the possibility of getting each of these gametes is random, right? There's an equal probability for all of those. And so the ratio, oops, if I can spell gametes, is always as so. Or if you want to write it out as 25%, it's going to be 25%, or in percentage, it'll be the same like that. It doesn't really matter which you use. Because the process which generates the random inheritance of alleles, right, which is, you know, encapsulated by the law of segregation, it's occurring for each of those loci, right? And so that means that each of those gametes is going to be present in, one presumes, roughly equal proportions, right? It's not going to be exact. And this is really where we're going to be getting into the chi-square test goodness of fit, right? Which you'll be doing maybe Wednesday, definitely by Friday, if not. And you'll also be doing this in the lab as well. So essentially what, kind of give you a heads up, this ratio is essentially the way of generating your expected values, right? So expect these gametes, right? Gametes are only, from one individual, only half of the equation. And so you need to add those gametes, oops, together, right, to get, essentially this is one parent up here on this side of the Punnett square. This is one parent down here on the other side of the Punnett square. You just add those gametes together, right? The gametes are the ones that are in the circles, essentially, ones that Mackenzie wrote out. So you add those together and then you look at what phenotypes you're going to get as a result. And because of, you know, the way it works, kind of depends on which way you want to put it around. But essentially you'll get your 9 to 3 to 3 to 1 ratio, right? So this is a kind of mathematical exercise, essentially. But obviously biology does not follow pure mathematics, right? It's a little noisy. And so depending on how many you count, right, in terms of what we talked about in the first lab, how big is your sample size? The bigger it is, the closer your goodness of fit should be, right? As long as things are working out to what you assume. Pardon me. You're going to get numbers which one hopes approximate this ratio. And then you're going to use this ratio as your expected, right? So you're going to be able to calculate what numbers do you expect. So let's say you counted 160 Ps, right? One would expect to find 90 that are round in yellow, 30 that are round and green, 30 that are wrinkled in yellow, and 10 that are wrinkled and green, right? Based on this phenotypic ratio, this is what you expect. And I chose 160 just because that makes the math a lot easier. Now you may have ended up with, say, 88, 32, 28, and 12, right, instead. And so you will be using, and this is what you'll be learning later on in the week, how to use the chi-squared test, goodness of fit, to see if those numbers are close enough to your expected, to this phenotype ratio, to tell whether or not that is essentially what you should be getting, right? Whether if that noise is of any meaningful import or not, right? And most likely, if your cross is doing what it should be, your assumptions are true, and you collected enough individuals, then you should get pretty close to the dihybrid F2 ratio of 9 to 3 to 1. And if you do, then even if it differs a little bit, then your chi-squared value will be pretty small and it will not be significant, right? So essentially, kind of getting back to this as a kind of a framework, this dihybrid cross is very useful because it gives you an expected outcome, right? If all of the assumptions that you started with, again, sex is random, no mutation, non-overlap in generations, no selection, right? And the law of segregation and independent assortment are occurring, i.e. alleles are inherited randomly or go into gametes randomly rather. And what allele you get for one gene does not affect what allele you get for another. Then you should get numbers that are pretty close to this ratio. Now, obviously, if the law of independent assortment does not hold true, right? That you know, the two genes that you're looking at are fairly close to each other on one chromosome, which may have been the case for some of the things that Mendel looked at, but he didn't know what that meant. Then you will not get a 9 to 3 to 3 to 1 ratio, right? You'll get ratios where these two in the middle essentially are lower than one would expect, right? Because the result would be the result of recombination, right? If these genes are interacting in other ways, right, such that the phenotype for one masks the phenotype for another, depending on which phenotype you get, then you'll also not get a 9 to 3 3 to 1 ratio, right? So that's telling you that something else is going on, right? So these are really, really useful kind of structures to use to be able to test whether or not genes are behaving in the way that they're supposed to. I don't know if I told you lot this or not. It kind of blurs between two different sections. But I spent a good number of years. Did I tell you this about a really awful cross that I had to spend years doing? I can't remember. It might have been the other section. Hopefully, it was the other section. See you later, love. But essentially, I wanted to get a GFP transgene and a mutation together to be able to look at the effect of the mutant or mutation on the localization of the GFP, right, where that fluorescent protein, sorry, GFP is green fluorescent protein, where that green fluorescent protein was located in the cells, and trying to figure out answers to questions as a result. And I pretty much never succeeded. I tried doing the cross 5 or 6 times, I think, over 2 or 3 years. And, pardon me, basically, whenever I did get the two things together, if they were homozygous, the worms either died or they're sterile, which is, in genetics terms, the same as being dead. Obviously not for the worms, they get to live for a little bit. But yeah, it was bloody shit show, to be honest. It was intensely frustrating. Kind of ended up getting it with one of them as being heterozygous, but then I had to maintain them as heterozygotes for the whole time, which was also a pain. So obviously, those two loci, where I don't even know where one of them was, were interacting somehow. Right? I don't know how. I still don't know. It's actually kind of curious, to be honest. But whatever it was, when both of those were homozygous, the worms died. So that's also clearly something funky is going on. It's not just me being crap at doing genetics. It's something is interacting there. And that's really dihybrid cross, right? So essentially, one of the reasons why, and there's a whole bunch of definitions down here which you should know because they're important. The whole benefit of Mendelian genetics is really not Mendelian genetics in itself, right? It's not the, well, I mean, one of the benefits is that it trains us how to think about inheritance in terms of probability, right? In terms of ratios and expected numbers. It kind of formalizes inheritance in a sense. But really its strength or power is how do we use that to look at things that do not follow Mendelian genetics or the laws of Mendelian genetics, right? And those are in the vast majority, right? Because obviously genetics is far more complex than the simple traits on individual chromosomes that Mendel studied. But without this kind of framework to work off of, these assumptions that Mendel gave us, it would be a real struggle to look at those interesting things, right? And so later on, this will probably be next week. Yeah, this is next week. Oh, no, it's not. I'm sorry. This is after exam 1. Anyway, you'll be studying exceptions to Mendelian genetics, right? Those will be things like, extensions of Mendelian genetics, such as, codominance, like red blood, your blood type, for example, is a good example of codominance. Incomplete dominance. You'll be studying things that are exceptions to it, which are things like epistasis, and so on, right? And so essentially, right, this is the ground level of inheritance or transmission genetics, and there's a whole bunch of other stuff built on top of that, but you obviously need to get that ground level first, right? So that's the real strength, in my opinion, of Mendelian genetics, but it's a small part of inheritance. And that's pretty much all I have for today. There's not a whole bunch more to talk about. Oh, question. That's a big question. All right, give me a second, Zuleika, to read that. So trying to understand your questions, Zuleika. So really a chi-squared test is a pretty formal test that can only be done pretty much in one way. And the way to do that is you're comparing your observed and your expected, right? So let me get back to here. Pardon me. So I'm gonna, where are we at? So here is the example that I gave. I think it was last week. So essentially, a chi-squared is you're adding together the sum of the differences between, or there's other stuff going on in there as well, between what you observe and what you expect, right? So essentially, what you're saying, what you're testing is what I observe within like a small difference of what I expect, right? And your expected values are based on assumptions, right? So for example, in a monohybrid cross like the one I'm showing, your expected values are based off of the phenotype ratio that you would expect in the F2 generation, right? Which is based off of crossing together heterozygotes in your F1 generation or from your F1s. Right, so you should always get a 3-to-1 ratio of dominant to recessive phenotypes in your F2 generation. So if you were to, for example, let's just type it out. So your chi-square, however you write that, is sum of observed minus expected. Oh yeah, that's squared. Divided by your expected. Right, and so to find out your expected, you take your total. So in this case, I got to do some math now. See, you're forcing me to do math before it's my time to do math. Let's see, what is that? 5474 plus 1850. Right, so your total is going to be what's that? 7324. Right. And then you by the fraction you expect. Let's say, what's that, 3/4 for dominant phenotype. Does that make sense? All right, so we're working out your expected. So at the present, let me see if I can get back and share my screen so you can see exactly what I'm doing. Oops, get off. There we go. So can you see what I've got up at the moment? Okay, cool. So, but the thing is, I've got to work through all of this, right? Because I can't just tell you part of it, because it won't necessarily make any sense, right? So you're expected for dominant. You'll have to forgive the shorthand. Is what's that? 7324 times 0.75. So that's 5473. 5493. And you're expected for yes, Lauren, it'll be exactly the same as what I've described already. I'm going to have to let Mr. Penny decide exactly what questions are going to be and what, because he's going to be the one teaching the class, right? So I don't know exactly what things he's going to teach you precisely and exactly what emphasis he places on them. And so I have to leave it up to him. I can't just give him the exams that I would give you, because essentially I don't know exactly what he's going to be teaching you, right? It's kind of a clunky way of saying I have to give him the freedom. Oops, shit bits. I have to give him the freedom to teach you the class in the way that he would teach that particular material. And as a result, that may end up emphasizing one thing more than another, or maybe he'll tell you one particular anecdote or something which he wants you to know and I wouldn't. But the actual layout, the format, how they're going to be held, you know, doing it on Blackboard, online, you having the day to do so, that sort of stuff, that will be the same. Okay, so these are your expected values. Your observed values are 5, 4, hang on, 5, 4, 7, 4, 4 dominant, and 1, 8, 50 for recessive. Right, and so essentially what you're going to do is you're going to do two calculations, and you're going to add them together to get your chi-squared value. So you're going to do for dominant, it will be, what is it? Observed 5, 4, 7, 4. Oops, minus, what's the expected? 5, 4, 8, 3. That's going to be squared divided by 5, 4, 8, 3, which is, what's that going to be? You always square it because exactly this issue, you have a negative number. So have that, and then you divide 81 by 5483. So that gives you a small number, which is what one would expect if things were doing what you expect, right? So for recessive, you do the same thing, but with the numbers for recessive, which, okay, they've disappeared up here. Let's just scooch this up a bit. It's going to be 1850 minus, where is it, 1831 squared divided by 1831. Sorry for the giant fingers, my camera's in a weird place. Makes it look like I have tarantula fingers. Right, so that's going to be 18, that, oops, sorry, typing it out in the wrong place. Get over here, 50 minus 1831. It's going to be 19. You square that, divide that by 1831 equals well, that's 0.2 basically. Right, now because this is the sum of those, you have to do this for every single group, right? You can have, if this were a dihybrid cross, it would be for dominant dominant, dominant recessive, recessive dominant, recessive recessive, right? So you'd have 4 different groups, right? So you have to add those together. So chi-square equals sum of, so that's going to be 0.015, oops, plus 0.2, right? What's that? 215. Now, the second thing you need to do is you need to calculate the degrees of freedom. And that's the number of classes minus 1. And that's not like how many classes are you taking right now. It's a bad joke. That's how many different calculations are you doing? We're doing 2, right? So it's going to be 2 minus 1 equals 1. Right now, I have to do a little bit of wandering around. Where are we? You get to see what I was reading before class. Exams and quizzes. Yeah, you logged me out after like the last 10 seconds. Come on. Here we go. Okay, so now what you do, and this is the key bit, you take degrees of freedom, 1, right, which is, you look at this chart here, right, and so you find the row that has your degrees of freedom in it, and then you take your chi-squared value, which is 0.215, and you see where that falls. Now, basically, it's way over here. Right? It's probably somewhere about 0.9 or 0.7, about where the University Police Department is, if that extended out that way. And so what that's telling you is that the probability level, right? The probability that you would get that result by chance, i.e. exactly what's happening should be happening, it's just a random fluctuation. The probability of getting that result by chance is very, very high, right? Like if you were to do this exact thing again, you would probably get the same difference, 70% of the time, essentially, right? And so p-value is more than 0.05. So the difference you saw between observed and expected is down to chance. Or another way of putting that is random, right? And so if it's just bound to chance or it's just random or whatever, that means that there's nothing wrong with your assumptions. So in other words, your assumptions are correct. Right, and so if you're assuming that this is where are we at? Go back here. You're assuming that this trait is behaving normally, that you have 2 alleles, that law of segregation applies, that you're actually doing a monohybrid cross, then you would expect to get a 3-to-1 ratio, right? And so by carrying out this chi-squared test, you're statistically testing whether that assumption is correct, right? Or those assumptions. And because you didn't get exactly a 3-to-1 ratio, but you got very close to a 3-to-1 ratio, the difference between what you observed, point, I don't know, 7.475 to 0.262 whatever, right? The difference between that assumption and what you actually got is small enough just to be complete random noise, right? Just biological cruft, essentially, right? And so you are able to statistically say whether or not that is the case. Now, if the results that you got with, let's say, what can we do here? Try doing this with, what is it going to be? 300 round, and get back over here. right? So try using different observed values, right? So try and repeat that chi-square test. This is essentially all the stuff that you'll be learning in lab and later on this week. But try doing that chi-square test again, exactly as I did kind of further up the chat session, and go to the exams and quizzes section of the lecture, and you'll find these chi-squared tables, and then see where your chi-squared values land. If the chi-squared value is smaller over here, oh yeah, this is going up on Blackboard as soon as I've started making a coffee and Zoom has finished doing its conversion process. This will go up within an hour or so. If your chi-square value is smaller than this number, i.e. it's on the left-hand side of this table, then the difference between your observed and your expected is not significant. It's due to random chance. If the number is bigger, right, which is what you'd expect, right? So if the gap between what you observe and what you expect is really huge, i.e. your expectations were wrong, then your chi-square value is also going to be bigger, right? And so the bigger your chi-square value, if it's larger than this value, it's essentially saying that the difference between what you observe and expect is not due to chance. It is due to something else going on which you have not accounted for, right? And so essentially, in the terms of a chi-square test and what we're talking about, that's saying that one of your assumptions is wrong, right? And so I don't know what that would mean with a monohybrid. One of your parents is not actually heterozygous, let's say. You know, it may be actually if it was a kind of going a little bit on a not that one. If one of your F1s was actually homozygous recessive, right, which it shouldn't be. Oh yeah, I'm sorry, Halia. Yeah, I'm sorry that I've run over. This was more just kind of like general chuntering on. Please go on to your next class. I didn't mean to keep you here. As same for everybody, and this will all just get uploaded. So anything that you've missed by needing to be somewhere else, just catch up on here. But yeah, essentially, you're assuming that these are all heterozygous. If one of them is not, then you're going to get a different ratio, right? And so that's going to essentially unmask that, right? That one of your assumptions is wrong, and then you go back and go, well, what is wrong? And then you start figuring it out. All right, I've taken up too much of your time already. So yeah, I'm sorry that essentially the homeworks are inherited, and I do mean to go through them and make sure they match up with the class material better. But sometimes they're a little bit ahead of what we're learning in class, I'm afraid. And sometimes they're a bit behind. It's kind of like wacky races, essentially. But yeah, and this is something that you'll be going through in lab this week, and also in lecture later on this week as well. So you'll be getting plenty of chi-square practice. Cool. All right, everybody. Yeah, hopefully I will see you again very soon. That would be yeah. Bye-bye, kitten. Super, super cute. Yeah, hopefully I'll be seeing you as soon as is possible for me to do so. But if not, the same applies. Study hard. Ask Mr. Penny any questions that you have as much as you would ask me, right? So please see him as being, you know, essentially me until I get back. All right. Have fun, everyone. Stay safe, and hopefully see you soon. Thanks, Jade.